The word blastula means sprout, a hollow sphere of cells called blastomeres, surrounding an inner fluid cavity called the blastocele, formed during an early stage of embryonic development in animals. Embryo development begins with a sperm fertilizing an egg to become a zygote, which undergoes many cleavages to develop into a ball of cells called morula. Only when the blastocele is formed does the early embryo become a blastula. It precedes the formation of the gastrula, in which the germ layers of the embryo is formed. The development of multicellular organisms begins from a single-celled zygote, which undergoes rapid mitotic cell division to form the blastula. During this stage of development, a significant amount of activity occurs within the early embryo to establish cell polarity, cell specification, axis formation, and regulation of gene expression. By the process of cleavage, the zygote divides into 2 cells that divide into 4 cells, then into 8 cells, and so on. Each division takes from 12 to 24 hours. With each successive subdivision, the ratio of nuclear to cytoplasmic material increases. Initially, the dividing cells called blastomeres are undifferentiated and aggregated into a sphere. When 8 blastomeres have formed, they begin to develop gap junctions, enabling them to develop in an integrated way and coordinate their response to physiological signals and environmental cues. When the cell's number is around 16, the solid sphere of cells within the zona pellucida is referred to as a morula. At this stage, the cells start to bind firmly together in a process called compaction, and cleavage continues as cellular differentiation. After the cleavage has produced over 100 cells, the embryo is then called the blastula, which has a spherical layer of cells called blastoderm surrounding a fluid-filled or yolk-filled cavity called the blastocele. In Xenopus, the animal cap forms the roof of blastocele and goes on primarily to form ectodermal derivatives. The equatorial or marginal zone, which compose the walls of the blastocele, differentiate primarily into mesodermal tissue. The vegetal mass is composed of the blastocele floor and primarily develops into endodermal tissue. In mouse, blastocele formation begins at the 32 cell stage. During this process, water enters the embryo aided by an osmotic gradient, which is the result of sodium-potassium ATPases that produces a high sodium gradient on the basolateral side of the trophectoderm. This movement of water is facilitated by aquaporins. A seal is created by tight junctions of the epithelial cells that line the blastocele. Mammals at this stage form a structure called the blastocyst, characterized by an inner cell mass or embryoblast that is distinct from the surrounding blastula, which has an outer layer, the trophoblast. Further compaction of the trophoblasts make it indistinguishable and is enclosed within the zona pellucida to make the structure watertight, since the cells later secrete fluid. The inner mass of cells that differentiates to embryoblast polarizes at one end and gives rise to the embryo proper, the amnion, yolk sac, and allantois. They close together and form gap junctions in order to facilitate cellular communication. In the blastula, these gadolinium-mediated cell interactions are essential to the development of epithelium, which are most important to paracellular transport, maintenance of cell polarity, and the creation of a permeability seal to regulate blastocele formation. Within the blastula, inner blastomeres are generally nonpolar. Mammalian embryos undergo compaction around the 8-cell stage, where cadherin as well as alpha and beta catenins are expressed, which helps in interaction of cells. E-cadherin addition defines the apicobasal axis in the developing embryo and turns the embryo from an indistinct ball of cell to a more polarized phenotype, which sets the stage for further development into a fully formed blastocyst within 5 to 6 days after fertilization. At this time, the blastocyst has reached the uterus, but has not yet implanted into the uterine wall. On day 5, pre-implantation human embryo contains 200 to 250 cells, only of which 30 to 34 are inner cell mass. In subsequent development, the cell of the inner cell mass give rise to all tissues of the embryo's body. As the size of the blastocyst then increases, it hatches through the zona pellucida, which then disintegrates completely, exposing cells of the trophoblast. This allows the blastocyst to attach itself to the endometrium for implantation. The formation of the two main layers of the bilaminar germ disc hypoblast and epiblast occur at the beginning of the second week. Either the inner cell embryoblast or the outer cell trophoblast will turn into two sublayers. The inner cells will turn into the hypoblast layer that will surround the other layer called epiblast layer, and these layers will form the embryonic disc in which the embryo will develop. The embryo plus its membranes together is called the conceptus, and by this stage the conceptus is in the uterus. The place where the embryo develops is called the amniotic cavity, which is the inside of the disc. The trophoblast develops into two sublayers, the cytotrophoblast in front of the syncytiotrophoblast lying inside the endometrium, and the exocoelomic membrane or Husser's membrane surrounding the cytotrophoblast, as well as the primitive yolk sac. The syncytiotrophoblast grows into the lacunar stage in which some vacuoles appear and is filled by blood in the following days. The development of the yolk sac starts with the hypoblastic flat cells that form the exocoelomic membrane, which coat the inner part of the cytotrophoblast to form the primitive yolk sac. An erosion of the endothelial lining of the maternal capillaries by the syncytiotrophoblastic cells of the sinusoids will form where the blood will begin to penetrate and flow through the trophoblast to give rise to the uteroplacental circulation. Subsequently, new cells derived from yolk sac will be established between trophoblast and exocoelomic membrane that will give rise to extraembryonic mesoderm, which will form cavities known as chorionic cavity. At the end of the second week of development, some cells of the trophoblast penetrate and form rounded columns into the syncytiotrophoblast, known as primary villi. At the same time, other migrating cells form into a new exocoelomic cavity named as secondary or definitive yolk, which is smaller in size than the primitive yolk sac. The epiblast gives rise to the fetus itself, while the trophoblast develops into part of the placenta and the primitive endoderm becomes the yolk sac. The trophoblast will contribute to the placenta and nourish the embryo. After ovulation, the endometrial lining transforms into secretory lining and prepares to accept the embryo. It becomes thickened with its secretory glands and increased vascularization. This lining of the uterine cavity is now known as decidua, and it produces a great number of large decidual cells in its increased interglandular tissue. The syncytiotrophoblast implants the blastocyst in the decidual epithelium by projections of chorionic villi forming the embryonic part of the placenta. The placenta develops once the blastocyst is implanted and connects the embryo to the uterine wall. The process of blastulation varies from different species to species depending mainly upon the amount of yolk present in the developing blastula. Blastulation in oligolecithal egg. The oligolecithal egg contain little yolk as in Amphioxus and Echinoderm. Holoblastic cleavage occurs in them and the blastomeres are not of equal size. The blastomeres at the vegetal pole are slightly larger than those on the animal pole. After the formation of blastula, the cells arrange themselves into a simple cylindrical epithelium enclosing the blastocele in the center. The cells from the vegetal pole of the egg form the floor of the blastula or hypoblast and form the future endoderm of the embryo. The cells from the animal pole region form the roof called epiblast and will give rise to the epidermis. Some cells from this layer form the prospective endoderm and form a ring around the blastula. This ring consists of two general areas: the dorsal crescent that give rise to presumptive notochord and neural plate cells, and the ventral crescent that gives rise to presumptive mesoderm. Blastulation in mesolecithal egg. Mesolecithal eggs have large amount of yolk and are present in animals like amphibians. The layer of cells surrounding the blastocele is of uneven thickness. The hypoblast cells lack pigmentation and are much larger than those at the animal pole and form the endoderm and later form the gut. They displace the blastocele to be acentrically located. The epiblast layer is several cells thick, darkly pigmented, and gives rise to future ectoderm, which become the future epidermis and nervous system of the embryo. The area of blastula consists of a ring of cells around the equator and forms the presumptive mesoderm. The notochord material forms just dorsally to the cells that first migrate into the interior of the blastula. Above this, the presumptive neural ectoderm forms the structures that give rise to brain and spinal cord of the animal. Blastulation in isolecithal egg. In animals like bony fish and shark, the cleavage is complete and the blastomeres are loosely bound together during the early stages, while they adhere to each other more firmly during later stages and convert into epithelium. The blastoderm develops only in the polar region and assumes the shape of disc called blastodisc on the animal pole. Blastulation in polylecithal egg. During meroblastic cleavage, as in fishes and tortoises, the blastoderm is single layered, loosely connected and is made up of one layer. Blastulation in centrolecithal egg. In insects, the cleavage is superficial and blastocele is negligible. The epithelium forms on the surface of the eggs from the blastoderm, which envelopes the mass of uncleaved yolk. Blastulation in microlecithal egg. Microlecithal eggs are those that have least amount of yolk, such as in true placental mammals who undergo holoblastic cleavage. The inner cell mass, however, spread out and form a plate or disc like those in bird or reptiles. The mammalian blastodisc divides into epiblast and hypoblast as in chick and reptiles. The hypoblast gives rise to the endoderm, while the epiblast gives rise to epithelium, neural ectoderm, notochord and mesodermal components of the embryo. Both the hypoblast and epiblast cells are considered as formative cells. The envelope of cells surrounding the inner cell mass become the nutritive cells or trophoblast. The lack of yolk in mammalian egg forces the embryo to find a nutritive source early in development and hence establish a relationship with the maternal body. The types of blastula vary a great deal among in different animals depending upon a variety of factors such as the size of the egg, the amount and distributional pattern of its yolk and types, the rate and number of cleavage division, etc. We can categorize them into the following types. Holoblastically formed blastula. In holoblastic embryos, the blastula is either solid or hollow. Coeloblastula. In such type, the egg divides by holoblastic equal division, resulting in the formation of blastula containing one or several layers of numerous cells arranged around a spacious blastocele. The cells may either be same or centrally placed blastocele as in echinoderms. Otherwise, the vegetal half has cells which are thicker than the animal half with a centrically placed blastocele. It occurs in hydra, amphioxus and frog. Stereoblastula. This type of blastula is composed of densely packed but larger sized and relatively smaller number of cells without or with extremely small blastocelic space in the center. It occurs in variety of animals such as insects, worms, mollusks, etc. Meroblastically formed blastulae. In meroblastic embryos, the blastula contains layers of cells over yolk. Discoblastula. It is formed by discoidal meroblastic cleavage. The blastomeres form a flat disc or blastoderm resting on the yolk mass. Underlying the blastoderm, the yolk is digested to form the blastocele, that is roof, has cells but floor has yolk. It is found in reptiles, birds and egg-laying mammals. Superficial blastula. This type of blastula is formed in insects and some other arthropods whose eggs are centrolecithal with a superficial type of cleavage. It does not have blastocele and consists of single layer of epithelial cells surrounding the centrally placed yolk. Other types of blastulae. Stomoblastula. In this case, an opening appears in the center of the blastula which functions as the mouth. It occurs in cycon. Blastocyst. This type of blastula is found in mammals. A cavity of blastocele develops during cleavage as solid morula, which gradually enlarges until it almost completely separates off an outer layer of cells, that is trophoblast, from an outer cell mass called embryonic knob. In the next step, series of morphogenetic movements occur in the blastula that locate the prospective tissues of the body in the appropriate places in structural plan to form a structure called gastrula, and the process is called gastrulation. It brings about orderly and integrated steps of reorganization and displacement of organ forming areas. The organizational center is established by the late blastula stage and directs the cellular migration during this period. The blastoderm gives rise to the three germ layers, the ectoderm, mesoderm and endoderm, and are formed as three overlapping flat discs. It is from these three layers that all the structures and organs of the body will be derived through the process of somatogenesis, histogenesis and organogenesis. The embryonic endoderm is formed by invagination of epiblastic cells that migrate to the hypoblast, while the mesoderm is formed by the cells that develop between the epiblast and endoderm. The cells normally migrate to the inside of the embryo along predetermined routes predictable for each species. In general, all germ layers will derive from the epiblast. The upper layer of ectoderm will give rise to the outermost layer of skin, central and peripheral nervous systems, eyes, inner ear and many connective tissues. The middle layer of mesoderm will give rise to the heart and the beginning of the circulatory system, as well as the bones, muscles and kidneys. The inner layer of endoderm will serve as a starting point for the development of the lungs, intestine, thyroid, pancreas and bladder. Following ingression, a blastopore develops where the cells have ingressed in one side of the embryo and it deepens to become the archenteron, the first formative stage of the gut. As in all deuterostomes, the blastopore becomes the anus while the gut tunnels through the embryo on the other side where the opening becomes the mouth. With a functioning digestive tube, gastrulation is complete and the next stage of neurulation begins. The blastula and gastrula stages are fundamentally similar in all animals. The particular shape of blastula is not important. At each cleavage, the chromosome with the genes are replicated and distributed to blastomere. The nuclei of blastomeres are identical. The general areas of the blastula give rise to certain structures. The blastulation process helps in the segregation of these major organ forming areas in a particular pattern. In the later blastula stage, a physiological center is established which organize and direct the migration of major organ forming areas. The rearrangement of blastomeres through morphogenetic movements called gastrulation give rise to the 3 germ layers out of which the future organs are constructed. The study of blastula and cell specification has many implications on the field of stem cell research as well as the continued improvement of fertility treatments. The field of embryonic stem cell has tremendous potential for treating disease. By manipulating the cell signals during the blastula stage of development, various tissues can be formed. This potential can be instrumental in regenerative medicine for disease and injury cases. The knowledge is generally used clinically in fertilization technologies. In vitro fertilization involves implantation of a blastula into a mother's uterus. Blastula cell implantation could potentially serve to eliminate infertility. Experiments with implantation in mice show that hormonal induction, superovulation and artificial insemination successfully produce mice embryos. These results prove to be encouraging because they provide basis for potential implantation in other mammalian species such as humans. Stem cells. Blastula stage cells can behave as pluripotent stem cells in many species. Pluripotent stem cells are the starting point to produce organ-specific cells that can potentially aid in repair and prevention of injury and degeneration. Combining the expression of transcription factors and local positioning of the blastula cells can lead to the development of induced functional organs and tissues. Pluripotent. Xenopus cells, when used in an in vivo strategy, were able to form functional retinas. Thus, at the end of cleavage, the embryo consists of a hollow sphere of cells called blastula. The blastula may be a hollow ball of blastomeres as in case of holoblastic types, or there may be layer of cells over yolk as in meroblastic type. The layer of cells is known as blastoderm while the cavity is called blastocele. The blastula eventually organizes itself into 2 layers, the inner cell mass which becomes the future embryo and the outer layer or trophoblast which becomes the future placenta. During gastrulation, the blastula folds in on itself to form 3 germ layers, the ectoderm, the mesoderm and the endoderm that give rise to the internal structures of the organism.